Hello, greetings to you all. This is Sushma Naithani from Oregon State University. First, I would like to thank Dr. Saurav Raghuvanshi for inviting me to this meeting. Today, I am going to share with you some of the insights from biocuration, particularly from viewpoint of plant pathway database curation. The need for biocuration stems from the availability of large amount of big data in the public domain that is continuously emerging from advanced high-throughput experimental data platform. Today, close to 100 plant genomes have been sequenced, including various model plants and crops. But the question is, where do we go from here? Sidney Brenner has put this thought few years ago. Getting the sequence will be easy part as only technical issues are involved. The hard part will be finding out what it means, because this poses intellectual problem of how to understand the participation of genes in the functions of living cells. The first challenge is how to deal with this data, basically data deluge, which means we have to develop the infrastructure for storage of data and consistent policies and standards for formatting various data types. Requirements for metadata and decisions about making this data public or private. And if it is accessible, how to make it findable using the concepts of semantic web and ontology. This process has been successfully accomplished for processing primary sequence data, and there are robust pipelines and dedicated efforts from past few years in that direction. However, various new type of data that are emerging, we do not have these things in place. We do not have, for example, any infrastructure or policies for making phenotype data available and stored in a public platform. Similarly, resources for many model organisms are well developed, but the efforts for crop plants are in initial stages. For example, in 2016, we put together a white paper listing needs of development of genomic and bioinformatic resources for Grapevine community. As you can see that various type of data from various international project is coming to public repositories, but we still need to build connections and relate one type of data to other type of data in order to understand grape biology. In addition to supporting species-specific communities, we need to facilitate comparative analysis across multiple species to understand evolution of life forms, biological processes, and unique attributes of organisms. To do so, researchers working on various organisms have to talk to each other. They have to agree on use of consistent data formatting standards and policies for biological data curation, on use of ontologies to describe various data types. Recently, major publicly funded databases working on various organisms of agriculture importance have developed a community portal, AgBiodata. So this is the website, agbiodata.org, and we are putting together a white paper for biocuration, data sharing, infrastructure needs, and policies. As we speak, these concepts, tools, and softwares are work in progress. For example, new ontologies itself are being developed by Gene Ontology Consortium and Plantium Project for plants. So for biocurators who are working with plant systems, they can find a lot of resources at plantium.org. This is the website of Plantium Project. So here you can find a lot of gene ontology related terms to describe various data type, but also specific ontology terms to describe plant anatomical structure, growth stages, and various plant traits. So you can browse along with this line and find various features that are here. So The first lesson of biocuration is that we cannot do everything alone. Doing any type of biocuration will require active international collaborations, training and retraining of workforce, including curators and software developers. Now I will talk to you about biocuration of plant pathways. First, I will talk to you about biopsych databases that represent metabolic networks in various organisms. These metabolic pathway databases were built on Pathway Tools software that was developed by Peter Karp and his colleagues at SRI International. Peter Karp and his colleagues at SRI International also developed reference databases for bacteria that was primarily curated based on published literature from E. coli, and it is called EcoPsych. They also created MetaPsych that was based on published literature gathered from various eukaryotic organisms, and it represents a generic metabolic network database for eukaryotes. So using the Pathway Tools software and reference database MetaPsych and EcoPsych, several groups have made species-specific metabolic network databases for many organisms for which the whole genome sequences became available. Here is a list of some of the plant metabolic networks that were developed by various publicly funded projects. This side is various dicot species-specific metabolic network. This side you have rice psych, sorghum psych, maize psych, and brachypsych that were developed by Grameen. I have been part of the curation of maize psych pathways. In my laboratory, we have developed Vitipsych, that is a grape-specific metabolic network, Eucalyptus psych, and Fragaria psych, in collaboration with Dr. Pankaj Jaiswal at Oregon State University. In addition to these psych databases, there are more than 1,000 psych databases available for various species of bacteria, fungi, and other eukaryotes. Altogether, all psych databases share the common features and tools, and so here I will walk you through using example from Vitipsych. So if you click here more info on any of these databases, you will land into the specific homepage of a given database. So this is the homepage of Vitipsych that serves as a springboard, and here you can access various different type of information about this database, as well as you can access the tools. So for example, if you click here on view summary, you can see that this database has about 430 pathways and close to 3,000 enzymatic reactions. You can also see the cellular overview, and I'll talk to you a little bit about what this cellular overview is. You can from this page, you can also upload your data here, any type of the gene expression data from grape, and you can analyze that data in context of metabolic networks. This is a generic view of pathways in psych databases. You have basically a substrate, you have a product, you have a reaction that is catalyzed by an enzyme, and that enzyme has mapping to various genes. Here you see the EC number of enzyme. In these databases, you can have entry of any metabolite only once, and therefore these metabolites are used to connect one reaction to another reaction, then these another reaction, and you make a pathway. Using similar connection, you can basically from this pathway page can jump into the pathway where this substrate is actually end product of the pathway. So you can go from one pathway to the previous pathway, or you can go to the following pathways where the end product of this pathway is being utilized for synthesis of other compounds. You can see also more detailed view where you can see chemical structure of each metabolite, and you click again and you go to the detailed page of that chemical. You see the published information and connections to the other publicly available resources such as Chevy. You can also click on reaction, you can click on genes or enzymes and so on. So this is not a static view as in textbook, but it is really a dynamic view, and you can connect to the literature, you can connect to the summary about each entity of the pathway. So this is just a cellular overview of these metabolic networks that are connected together using common metabolites. In a simple virtual cell, you have biosynthesis related pathways on left side, you have degradation and assimilation pathways in the middle, and then on the right side you have various small reactions, you have various proteins or enzymes for which we don't know where to place them here. So here a lot of biocuration and new information that will come through various new experiments will help us to associate some of these proteins and enzymes to this organized metabolic network. Again, you can zoom in and zoom out, and you can actually see what these various lines and these dots means in the cellular overview. You can again jump from here to any detailed page describing either a reaction or any entity. So again, enzymes in YT-psych or any psych databases are grouped together based on their EC numbers and enzyme ontology. Similarly, if you see pathways, pathways are grouped based on connections in super pathways, subpathways, and here you have the whole hierarchy and listing here. You can browse any of the given pathways uh, using this schema view, and you can access any pathway here by clicking on this particular schema. You can again access information on reaction, about the chemical structure. You can get also the literature references right here. This is again a summary of how different genes are placed in different chromosomes and where do or what different type of the pathways basically you have. You can have also analysis tools here, and you can have comparative analysis. You can access various analytical features from any pathway detail page. So here, this right hand side, you can download these genes in two different formats. You can also upload omics data and overlay on this pathway, and then go to cellular overview and also from one pathway to another pathway. In comparison operation here, you can compare pathways across two species or multiple species. So for example, if here you click on species comparison pathway, you can see the list of the available database right here on this site, where you can compare pathways from one organism to another organism. So here I have chosen to compare Vitis pathways with Arabidopsis, and this is the output I get. I get all the gene mappings, their EC numbers, and their annotation right here in Arabidopsis, and I can see that also in Vitis. And as you can see, that some of the information is available for Arabidopsis but not yet for the Vitis. Sometimes we can use this information and look for the Vitis genes that share homology with these Arabidopsis genes, and then fill these gaps in the path. So this is where also a biocurator's role becomes very essential to identify the holes in the databases and bring new information that has not come through the automatic projections. One can also upload expression data sets and conduct analysis in the context of pathways. So this is just here is the output of the gene expression data that we have uploaded, and as you can see, high expression is denoted by yellow and red, and low expression is towards the green. So you can see in a particular sample, some of the genes are upregulated, others are downregulated. You can click on these pathways, or you can have a list of pathways where genes are upregulated or downregulated, so you can access this various formats. Before I show you that, I just wanted to show that you can upload many samples together, data from many samples together, and then you can go back and forward from one sample to another using these forward and backward arrows. Here is just an example of a pathway detail view and mapping of this expression data on phenylpropanoid biosynthesis pathway. As you can see, this pathway is very big, so you don't see detailed name of the enzymes and gene mappings right here, but you can go into more detailed view and access that information. Anyway, what I wanted to bring to your notice that here we have 4 samples, and so these are relatively unripe grape berries, and here you have ripe fruit and pink fruit right here. And so this is the RNA-seq expression data from this from one of the laboratory of my collaborator Laurent Duluc at Oregon State University. And as you can see that although you have multiple homologs present in whites for the same enzymatic function and it is mapped to the same enzyme, not all the genes, not all the homologous genes have the similar expression profile. Some of these are expressed early and in high amount, and others are not at all expressed, and others are expressed, as you can see, in a lower amount. So this analysis is a great tool for researchers to identify candidate genes which express in a given stage or in a given specific tissue types or growth stage or in response to various abiotic and biotic factors. So using this data-driven analysis, users can formulate a more plausible hypothesis for their experimental research and reduce the load of their experiments. So here, before I forget, I like to acknowledge my undergraduate student, Eli Weddell, who put a lot of work into curation of white-side pathways. And I would also like to acknowledge Pankaj Jaiswal, Rajni Raja, and Justin Elser from Jaiswal Lab for helping us to build this database. I would also like to acknowledge startup funds that I and Pankaj got from Oregon State University. I'm also thankful for Peter Carr for developing this wonderful tool and making it accessible to the community. Now I move to third part of my talk, that is about Plant Reactome. So Plant Reactome is a plant pathway portal of Grameen database, and it is funded by National Science Foundation. Here is our little team of Plant Reactome. Pankaj is the PI of Plant Reactome and Justin is software developer. Myself and Parul are mostly the curators, and then we have sometimes graduate students and undergraduate students who work for a little bit with the data. Before I go into details of Plant Reactome, I like to talk to you about data model that we use for curation of pathways in this database. So we use Reactome platform for curation of pathways. It was developed by Human Reactome Project. Reactome platform have several advantages. First of all, here is a depiction of a simple reaction. So you do have input molecules and output molecule, but here you can have any type of reactions. You can have, of course, metabolic pathways that are catalyzed by enzymes, and you can use a similar gene ontology and enzyme functions to describe and organize those pathways. In addition, you can depict any other type of event. It could be regulation or genetic regulation of any event via a protein or via RNA molecule, for example. It can be a transport reaction from one compartment to another compartment. It can be a transformation event. It can be dimerization, phosphorylation, and so on. So there is no limit of the type of the reactions that we can host using the Reactome platform. Again, we try to link our database with other public resources that provide complementary information on the structure of proteins or molecules or complexes or regulatory RNAs or disease variant or anything else. So we believe great in terms of sharing the data, reusing data and policies that has been developed by other public resources, and we are open to interoperability fully. We use control vocabularies for describing all the entities and pathways and events that are in our database. All the names for pathways, all the reactions and molecular entities, they have unique and unambiguous identity in our database that help us to make our pathways consistent and readable by machines, and this data then can be read by APIs and can be imported to the other public resources or to build other type of interaction networks as well. So we are open. We provide this curated data uh, is open for all, and we provide various ways by which users can access this data. Users can access planetary ectome database directly from Grameen homepage. As you can see that here are various portals of Grameen database, and planetary ectome is right here. So if you click on planetary ectome, you can get into planetary ectome. You can access planetary ectome from directly from planetaryectome.grameen.org. Again, here like Grameen, you can conduct search for gene, pathway, or any entity. You can also access analysis tools from here or access our data. Before I go into detail, I would like to provide you a summary what we have in planetary ectome. Currently, we use rice as a reference species for pathway curation and then use gene orthology-based methods to project these pathways on other 74 plant species. We update a Grameen database and planetary ectome database regularly. So in planetary ectome, if you click on content, you can find the recent release summary, and you can find about the statistics about number of pathways, genes, reactions, and so on, and also number of the species that we have data here. Here is a quick tour of the pathways and contents we have in planetary ectome database. So this is the pathway browser view. By default, we have reference species Oryza sativa listed here. This left-hand panel lists number of pathways and subpathways which are based on ontology concepts right here. On this right-hand side window is the pathway diagram window. Depending on the selection that you have made here, this diagram will change. So currently it is showing all the metabolism and regulation related pathways right here. You can click either in this window, or you can again open these tabs and go into details of the pathways and subpathways. This bottom panel here has associated information about pathway summary, about the various molecules depicted in any pathway, also options for downloading this data, and so on. So this is again the pathway detail view. As you can see from this list that earlier we have concentrated on curation of metabolic pathways. These pathways primarily were imported from rice site that Grameen has developed earlier and were adapted to reactome model. So a lot of biocuration has gone also in adaptation and creating their pathway diagrams and utilizing some of the previous space that we had from rice site. Recently, our focus has become more into other type of pathways that are genetic regulatory pathways, pathways related to plant growth and development, and how plant respond to various stress conditions and so on. So as you can see that we have here depiction of this regulatory pathway right here. In the bottom side panel, depending on the selection you make, so for example, you can choose to see if there is any expression data here, and here we can automatically pull down the basal expression data for rice that is made available remotely from EBI. Here is example of recently curated pathway related to root. You can see here I have highlighted ARF12, one of the transcriptional regulation that inhibits that is inhibited in response to phosphate deficiency, and then it further blocks the lateral root development. So we can see connection of various metabolic pathways and gene regulatory pathways, and we like to build connections with abiotic conditions and plant growth and development. This is another pathway that describes in detail response to phosphate deficiency, and again you can see that here we can find ARF12 that is one of the factors that affects the root development. There are other uh, players here, and they affect the different plant parts as well. Another example is also response to submergence. So this is a small part of response to submergence. What happens to shoot development and internode elongation in response to submergence. So here again we have genetic regulatory pathways, two main transcription factors, sub A and sub C, right here in this diagram, and then other components. And then we have genes such as SPL14, which gives the ideal plant phenotype and has a lot of importance in breeding. And then here we try to connect the hormone signaling as well as the important traits associated with rice. Again, if you pay attention here in this diagram, you see these number 2 here in red circle, and this is again a recent development. We are able to bring publicly curated gene-gene interaction data through API to some of our pathways here. So currently we have it available for rice and for Arabidopsis. So this is an example of a projection of gene-gene networks on pathways. So for example, using this gene that we have curated here in one of the pathway, we are able to bring another 48 genes that were brought from the ARANET interaction development, in ARANET gene-gene interaction database. So as I said that there are very limited amount of resources that we have for curation, especially for manual curation. We have only one full-time curator for plant reactome database. So we like to use and reuse and link to other resources and provide the best services we can provide to the plant community. So having extra nodes are always advantageous for our users so that they can formulate a more data-driven, more plausible hypothesis. Having gene-gene interaction data for any species will be useful. So if you have any good data and you would like to share with us, we will be happy to host it for your organism of choice or for the plant species that you are working on. So please contact us. Again, just a little bit about analysis tools. So here we provide two type of analysis tool. So if you say species comparison, you can choose the species of your interest and compare projected pathways with the reference pathways. You can also analyze your data and you can paste any type of gene expression data, metabolomic or proteomic data. This is just an example of the output, and here as you can see that gene expression data was overlaid on pathways for genes that found the match in our database. And so you can see the green color for low expression and red color for high expression. As we know that plant has multiple homologs for the same enzyme function. So you can click on the enzyme box here and you can see mapping of homologous genes, and you can see the differential expression profile of these genes and identify candidate genes for breeding purpose or other experiments that you like to conduct. So again, this feature is very useful for formulating a more data-driven hypothesis for experimental purpose. Not to mention, our data is fully accessible using APIs and in other data formats as well. I would like to acknowledge funding from National Science Foundation, Grameen colleagues from both Oregon State University and Cold Spring Harbor. I would also like to thank our collaborators from Human Reactome team, especially Peter Pistachio, who is our curation mentor. I would also like to acknowledge help from EBI and for their expression data projection that we provide on plant reactome. Also all data providers and other collaborators and Grameen users for their feedback and their support. Thank you very much for your attention. If you have any questions or any queries, you can write to me at nathans@science.oregonstate.edu. Thanks again.